Thank you. Good morning. My name is Sarah Foster. I'm an undergraduate here at William & Mary and Dr. Caitlin Barrett-Moore's research group. And today I'll be discussing our research on the application of GC GC for the investigation of fermented beverages. In our lab, we mainly focus on the use of comprehensive two-dimensional gas chromatography, or GC GC, for the separation of volatile organic compounds and identification. Our group really focuses on GC GC for applications like the forensic sciences and food sciences. For forensic science, we're doing a lot of probing different evidence types like fingerprint residue, gunshot residue, pepper spray, and more. And for the food sciences, we really focus on fermented beverages or foods that claim health benefits. Another large focus of our research is bringing the technique of GC GC through from proof of concept forwards towards its actual end use in the field, and that includes how data is presented and interpreted. When dealing with complex non-targeted data, there are a number of challenges to overcome. First, we resolve a lot of complex coevolutions. This means that we're seeing more of what's in our sample, and that more can sometimes feel overwhelming or hard to characterize. Additionally, because we resolve all of these coevolutions, we're seeing more features in our samples and accordingly an increase in the number of compound classes. Likewise, because we're able to separate lower level analytes from more saturating compounds, this affords us the ability to compare subtle differences between our samples. Additionally, the most abundant compounds are no longer necessarily the most important, and those low, more trace level analytes can really tell us a lot about our sample groups. And while this is all great for seeing more of what's in our sample, it can sometimes make longitudinal tracking much more challenging. To deal with these challenges in our lab, we have access to the full suite of software that LICA provides with their GC GC TOFMS systems, including Chromatoph and Chromatoph Sync 2D, and we supplement this with open access programs like our statistical software. For Chromatoph, we really use it for a lot of initial data processing. It's good for peak detection and deconvolution for single chromatograms at a time, and good for compiling peak tables for one chromatogram at a time. But often our sample sets have hundreds of chromatograms in them, so we want to do something a little bit more efficient, and that's when we use Chromatoph Sync 2D. Sync 2D is an alignment software that does peak detection and deconvolution for all of the chromatograms in your data set at once, and it gives you one peak table list for an entire data set, which is really useful when you're trying to identify trends across your data set. Meanwhile, we like to put all of this data into our statistical software for multivariate visualization. R is a really great software because there's a lot of programs or codes online that are easily accessible and free, which means that they are very customizable to meet the needs of individual experimenters. Because each data point has an associated primary retention time, secondary retention time, and intensity, dimensionality reduction techniques can be really helpful in helping to highlight the relationships between sample groups. The three main techniques that we employed throughout this project were principal component analysis, or PCA, principal coordinate analysis, or PCOA, and hierarchical cluster analysis, or HCA. All of the figures that we generated using these techniques were made in our statistical software using the vegan package and the complex heat map package. First, looking at principal component analysis and principal coordinate analysis, these are two very similar techniques that are based on slightly different mathematical principles. So they both use a dissimilarity matrix, which is essentially a square matrix that makes pairwise distances or distinctions between a set of objects. PCA operates directly on the data matrix using a Euclidean dissimilarity matrix, meaning that it assumes the variance structure in the data can be described by a linear trend. Meanwhile, PCOA uses non-Euclidean dissimilarity matrices, such as Bray-Curtis dissimilarity, and this is really helpful when the variance structure in the data cannot be exactly described by a linear trend. The basis of clustering for PCA is shared analytes, meaning that two groups that are close together on a PCA plot share a lot of similar volatiles. PCA is also really great for identifying key analytes that are driving variance in your data set. Looking at the PCA loadings plots can help you identify why certain groups on the PCA plot separated the way they did. PCOA comparatively looks at overall dissimilarities. So it looks at a lot of compositional differences, meaning that the PKOA plot is really helpful for describing similarities and dissimilarities in overall composition. HCA, or hierarchical cluster analysis, can use a Euclidean or non-Euclidean dissimilarity matrix based on the variant structure in your data. We use a Euclidean matrix for all of this. The basis of clustering is presence, absence, and a relative abundance of analytes, meaning that two sample groups have to contain the same analyte or lack the same analyte in similar quantities in order to cluster closely together on an HCA plot. The output of an HCA is a dendrogram, where shorter branches indicate more similar samples and longer branches indicate more dissimilar samples. The goals of this project were to explore the application of GC GC-TOFMS for the volatile analysis and aroma profiling of mead products, and to investigate the impact of different yeast strains and honey concentrations on the final aroma profile. We were looking at this through a non-targeted lens, and we were hoping to extract and compare features to make meaningful conclusions about our sample set. In general, mead is a fermented beverage made from honey blended with filtered water. Fruits and spices are added during the production for flavorings, and oftentimes the mead is aged in a barrel after it's been fermented. It can also be fermented to the brewer's desired level of alcohol content. In the experiment performed by our collaborators at Silverhand Meadery, 1-gallon batches of mead were made by blending honey with filtered water. They used 5 different dry yeast strains, which they rehydrated in filtered water for 20 minutes, and then they equilibrated the yeast to the mead by essentially putting little portions of the mead into the yeast. And then they pitched all of that back into the main gallon and allowed it to ferment. They did staggered nutrient additions on days 3, 5, and 7, and then they allowed it to ferment for a total of 9 months before filtering out all the yeast sediment and adding potassium sorbate and potassium metabisulfite as stabilizers. 5 different yeast strains were tested at 3 different honey concentrations. The first strain was Kveik Voss, which is a Norwegian yeast typically used to brew ale. It produces relatively neutral aromas, but can also have slight orange and citrusy notes. The next yeast was Munich, which is used to produce German wheat-style ales. And this yeast is known to produce higher order esters and phenols, giving it a banana, fruity, and clove aroma typically. The next yeast was EC1118, which is the yeast used to brew sparkling wines, and this typically has a very neutral aroma. Now looking at D47, this is a yeast used to usually brew white wines, and it has a tropical or white floral aroma. Lastly, they looked at Abbey, which is a yeast typically used in Belgian-style ales, and it produces tropical, spicy, and banana aromas at low temperatures and date, raisin, and fig aromas at higher temperatures. They also used 3 different starting concentrations of honey throughout this experiment. The first one was 1.5 pounds per gallon. This is known to produce a hydromel or low alcohol mead, and it was thought that most of the yeast strains under these conditions would finish dry, meaning they would be able to consume all of the sugar. The next condition was 3 pounds per gallon of honey, and this is the typical conditions under which mead is usually brewed. And here it was expected that a lot of the yeast strains, or some of them, could be challenged, meaning they would be unable to perform at their metabolic optimum. The last condition was 5 pounds per gallon of honey, and this was supposed to produce a sack or high alcohol mead, and all of the yeast strains under these conditions were expected to finish sweet, meaning they would be unable to consume all of the sugar. The honey used to produce all of these meads was the orange blossom honey from Silverhand Meadery, and the filtered water shown at the bottom is the filtered water used to actually brew the meads. The colors in all of the cases, the lightest color corresponds to the lowest concentration of honey, and the highest color corresponds to the highest concentration of honey. We looked at the mead samples, some of the plain honey, and then also some filtered water to use as a control. To prepare the mead samples for analysis, we aliquoted 3 milliliters of mead into a 20 ml headspace vial, added half a gram of salt, and 5 microliters of 500 ppm 1-decanol internal standard. For the honey samples, we combined 1.5 grams of honey with 1.5 milliliters of filtered water. We again added half a gram of salt and 5 microliters of the 500 ppm internal standard. For the filtered water, we just did 3 milliliters of filtered water with half a gram of salt, and again, the internal standard. We vortexed all of these and put them on our GC GC-TOFMS for analysis. Our GC GC-TOFMS is the LICO Pegasus PTX4D with dual TOFMS and FID detection. It has a reverse fill flush modulator and a splitter to divert the flow to both the TOFMS and the FID. This is what the modulator and splitter look like in real life. The modulator functions by filling a small sample loop in the forwards direction via the use of valves and back pressure regulation. Then, during the flush mode, the valves switch, sending the effluent onto the secondary column. This requires quite a high flow going onto the secondary column, more of which is capable of being handled by a TOFMS generally. So we split the flow, sending some of it to the TOFMS, and then the rest to the FID for additional analysis. Looking at some of the outputs using this method, on the top chromatogram is the orange blossom honey, in the middle is some filtered water, and on the bottom is one of the mead samples. The 1-decanol internal standard is labeled in each of them, and some of the other important components that we identified are labeled. For the honey, we identified furfural, which is a common volatile found in honey, especially honey that's been heated like it would be in the mead production process. In the filtered water, we found 2-ethylhexanol, and in some of the mead products, we found 3-methylbutanol and benzaldehyde, both of which are common fermentation products. Now getting into the results a little bit, for all of the following analyses, the honey and filtered water were removed in order to better visualize the relationships between the mead products. Here are the PCA results, and we can see kind of 4 groupings here, which were largely dictated by honey concentration. The first grouping is the 1.5 pounds per gallon groups of mead, which show up on the far right on the PCA plot. And here we can see that the D47 strain and Kveik-Voss strain completely overlap, meaning that they're essentially indistinguishable based on their volatile profiles, according to these PCA results. The EC1118 strain also grouped relatively closely to them, and then the Munich strain kind of separated up a little bit. The next group is the batches of mead brewed with 3 pounds per gallon of honey. And here we can see that the meads grouped a lot closer together, meaning that they have relatively similar volatile profiles. Here, interestingly, the Kveik-Voss strain kind of separated from the rest of them. Looking at the 5 pounds per gallon groups of mead in the bottom center, we can see that it's very close to the pattern we were seeing with the 1.5 pounds per gallon meads, where the D47, Kveik-Voss, and EC1118 overlap, and the Munich yeast kind of separates out. Interestingly, the groups of mead brewed with the Abbey yeast actually formed their own separate group over here. We can see that the groups of mead brewed with the Abbey yeast and 1.5 pounds per gallon of honey and 3 pounds per gallon of honey are very close to the overall 3 pounds per gallon group, but based on their positioning on the plot, it's relatively clear that the groups of mead brewed with the Abbey yeast had a more distinct volatile profile, at least one not as dependent on honey concentration. Now looking at the PCOA results, they're very similar to the PCA results, but there are a few differences. So first, the groups of mead brewed with 1.5 pounds per gallon of honey are in the same relative position, and the strains have the same relative relationship. The same can be said for the 3 pounds per gallon of honey meads. The confidence ellipses are a little bit larger, but they're all overlapping. So again, same conclusion that they share a lot of volatile similarities. Again, nothing really changed with the 5 pounds per gallon of honey group from the PCA to the PCOA, and they have the same relative relationship to one another. But the groups of mead brewed with the Abbey yeast are here clearly a more distinct group from the other batches of mead. And this could possibly be due to the fact that the PCOA uses Bray-Curtis dissimilarity, or a non-Euclidean dissimilarity, meaning that it's able to emphasize those compositional differences that PCA isn't really able to. Looking at the HCA results, this HCA was combined with a heat map analysis, where every row represents an analyte and the color represents the relative abundance of that analyte, with red being most abundant and dark blue being absent. The first grouping we saw here was on the far left. It's the groups of mead brewed with 1.5 pounds per gallon of honey. And here we can see that D47, EC1118, and Kveik-Voss are all tightly clustering. while Munich moderately clusters with the other three strains, which is exactly what we were seeing in our PCA and PKOA plots. So that's cool. And then on the far right of the plot is the groups of mead brewed with 3 pounds per gallon of honey. And here again, we're seeing tight clustering between all of our strains, emphasizing the fact that they are very compositionally similar to one another. And that can be also confirmed by the heat map. We see that they contain very similar volatiles in very similar concentrations. Now looking at the groups of mead brewed with 5 pounds per gallon of honey, we can see that D47 and EC1118 tightly cluster, along with Kvike-Voss, and then the Munich again moderately clusters with the other three strains, which confirms what we were seeing in our PCA and PKOA plots. Interestingly, the group of meads brewed with the Abbey yeast are again their own distinct grouping on the HCA plot. They are loosely clustering with the 3 pounds per gallon of honey groups, which again is what we were seeing in our PCA plots. Here, the 1.5 pounds per gallon of honey and 3 pounds per gallon of honey groups tightly cluster, which makes sense based on their positioning in the PCA and PKOA plots. And looking at the heat map, we can start to maybe understand why the volatile profile was so different for the Abbey yeast than it was for all of the other yeast strains. It seems to contain maybe similar volatiles, but definitely in lower relative abundances. So that might start to explain the separation of that strain of yeast from the other ones. We also saw 2 overall clusters in this data set. The first one was the batches of mead brewed with 1.5 pounds per gallon of honey on the far left, and then all of the other meads kind of formed their one big group. And this is exactly what we're seeing on our PCA and PKOA plots, where the 1.5 pounds per gallon of honey batches of mead were far right on the plot, and then all of the other groups were on the left or in the bottom center. We were able to detect a total of 96 volatile organic compounds in all of our mead samples. And when going through the data in sync, we noticed that there were quite a few trends in the volatiles that seem to correlate to honey concentration. We're still working on this to see if it relates to any other factors like compound class, but we wanted to show some of the initial patterns we were seeing. First, on the top left is ethyl decanoate. And here we can see that the relative abundance of ethyl decanoate increases as honey concentration increases. For furfural, which is again a common compound found in honey, it of course has the highest relative abundance in the honey itself, but also increases in relative abundance as honey concentration increases, as we would expect. Next, 3-methylbutanol, which is again a common fermentation product, decreases in relative abundance as honey concentration increases for all of the strains except the Abbey strain, where we can see that the 3 pounds per gallon mead actually had the highest relative concentration of 3-methylbutanol. We're not really sure why this happened, but it's interesting and potentially important for understanding why the volatile profile for the Abbey yeast was quite different from the other ones. Lastly, looking at ethyl octanoate, we can see that it had a little bit of a different pattern, where it peaked in relative abundance at 3 pounds per gallon, was the lowest at 1.5 pounds per gallon, and was kind of in the middle at 5 pounds per gallon. And we're not quite sure why this happened. We're still trying to explore that, but yeah. In conclusion, GC GC-TOFMS was a very effective strategy for the analysis of mead products and aroma profiles. We were able to use dimensionality reduction techniques to help highlight the differences in relationships between sample classes. And we're hoping to put more research into how the environment of microorganisms like yeast impacts their volatile organic compound production and how that would impact the final aroma profile or taste of the mead products. In the future, we're also hoping to connect the microbial kinetics data that Silverhand Meadery collected while they were brewing this mead to the volatile profiles we've developed here. I'd like to thank Dr. Kate and all of my lab mates for all of their support. Thank you to Silverhand Meadery, our collaborators, for helping us out with this project. If you'd like to taste some of their mead products, you can go to the mead tasting and Lico dinner tonight at 6:30. I'd also like to acknowledge the Arnold and Mabel Beckman Foundation for their funding that allows me to do research during the summer and academic year. Thank you so much for your time and attention, and I'll take any questions.